Wait, what?
Majority of stuff.
Micro?
Microanatomy, gross anatomy.
Oh, fuck.
Okay, so the first lecture on lipolysis. Okay, so insulin is the main hormone in the fed state, and what's directly opposite is going to be glucagon. So insulin fed, glucagon fasted. So you just need to know this in detail. Just know that insulin makes more GLUT4. And then for the enzymes or for the actual pathway, you just need to know the regulated steps and then the regulation of the regulated steps. And like the big concept is for regulation, substrates promotes products inhibit. And the only funky regulation one is for BFK, and it's with fructose 2,6 bisphosphate. I'll go more in depth on that. So just on the regulated steps, so hexokinase, BFK1, phosphoglycerate kinase, and pyruvate kinase. And then the other special one you need to know is it's the glyceraldehyde phosphate dehydrogenase. Hexokinase. Just know that this step is targeted by arsenate. And arsenate, it acts as a poison in glycolysis by mimicking a phosphate. So, see this? This is where arsenate goes. And what will happen in the next step is that you will not have ATP production. Just on this step, that's what makes ATP. And it's also where you make 2,3-DPG. And something that I also forgot to mention, when you block an enzyme, everything after that enzyme will decrease, everything before it will increase. So a good sign that there's a lockup is that if there's an increase in 2,3-DPG, because that's like the side pathway when there are, these enzymes here are too slow. Any questions about that? Okay. Good. Okay, this is pyruvate kinase. Just know that excess fructose 1,6 bisphosphate promotes it, because again, it just shows that there's a buildup of substrates. And then remember, substrates promote. So you need to know your sources of pyruvate and acetyl-CoA. So the sources of pyruvate are amino acids, lactate, and glucose. The sources of acetyl-CoA are amino acids, fatty acids, and pyruvate. So when there's no oxygen, you go to anaerobic glycolysis. So the function of pyruvate is to remake NAD plus in anaerobic metabolism. Okay, and then the enzyme that remakes the NAD plus is lactate dehydrogenase. This is updated so you can delete the slide. Oh, and fructose enters glycolysis through glyceraldehyde and DHAP. Galactose enters glycolysis through glucose 1-phosphate, and then that gets changed to glucose 6-phosphate. Whatever. Oh, and feed forward was the thing that I've been talking about. Products, inhibit, substrates, promotes. Hexokinase has a lower Kn, glucokinase has a higher Kn. And because glucokinase is in the liver, hexokinase is everywhere else. Okay. Let's see. Okay, just know that fructose 2,6-bisphosphate promotes glycolysis, but if it's fructose 2,6-bisphosphatase, it inhibits glycolysis. Any questions about that? Okay. So the enzyme that makes fructose 2,6-bisphosphate is PFK2, and that only, it's PFK2 when the enzyme is dephosphorylated, and that's done by insulin. So insulin dephosphorylates, making PFK2. So when this enzyme is phosphorylated, instead of becoming PFK2, it becomes fructose 2,6-bisphosphatase. And that's done by glucagon or epinephrine. Does anyone need to repeat that?
Yes.
Okay. So I'm talking about this enzyme right here. So when it's dephosphorylated, it's PFK2, and PFK2 makes more fructose 2,6-bisphosphate. And that's done with insulin. Okay, and then this one, it's phosphorylated, so it becomes fructose 2,6-bisphosphatase. And that's done by glucagon or epinephrine. So yeah. Everyone got that? Okay. One more thing about PFK1. PFK1 has 2 binding sites for ATP. The competitive site and then the allosteric site. So the competitive site has a lower Kn, the allosteric site has a higher Kn. So competitive site promotes it, allosteric inhibits it. Does this make sense?
Say that one more time.
Yeah, the competitive site has the lower Kn. So this is where the actual reaction happens. And then the allosteric site is the regulation site. So when there's high ATP, because remember ATP is a product, it will inhibit this enzyme. I think that's it. Yeah. So muscle does not have receptors for glucagon. Instead, it uses epinephrine. Any questions about this lecture?
I have a question but epinephrine doesn't really inhibit glycolysis like glucagon does, correct? In muscle.
In muscle, I would say it inhibits it. It inhibits glycolysis in muscle.
So you want the ATP to be produced
So the point of glycolysis is to make pyruvate, to make pyruvate, but you have other sources of pyruvate, or acetyl-CoA. Does this make sense? So instead of using glucose, you're using other sources like fatty acids or amino acids with epinephrine. Because you want to conserve glucose when you're using epinephrine.
I was thinking that it inhibits glycolysis in cells in tissue that's not muscles. It inhibits glycolysis in other cells, and then it sends it to the muscle.
Are you asking if epinephrine inhibits glycolysis?
In muscle, or it doesn't affect muscles.
Yeah, no, epinephrine inhibits glycolysis everywhere. Yeah, just don't worry about that, just know that.
Okay.
Because when you're in fight or flight, you're not focusing on building muscle, building stuff, right? You're
But you
just
need
focusing
energy.
on.
But you need the energy to.
Yeah, so you get energy from other sources besides glycolysis.
Okay.
If you still have trouble, just
Okay.
talk me after. Yeah, any questions?
Just repeat the PFK allosteric thing one more time.
Yeah, so PFK has two binding sites for ATP, the competitive site and the allosteric site. So the competitive site has a low Km, so that's where the actual reaction happens. And then the allosteric site is the regulation site, so that means a high Km, so high amounts of ATP will inhibit the enzyme. Okay, so TCA cycle, just know arsenites inhibits lipoic acid. I believe that's E2. Wait, this TCA cycle, okay. And whatever. Actually, okay. And when just know, again, just know the important reaction steps for this cycle. So anything with the dehydrogenase makes an NADH, and just know succinyl-CoA synthase makes a GTP. And succinate dehydrogenase is complex 2 in the ETC cycle. The anopyrrotic reaction, which makes oxaloacetate from pyruvate at the cost of 1 ATP, is done by pyruvate carboxylase, and pyruvate carboxylase requires Biochem. And the main regulator for the TCA cycle is pyruvate dehydrogenase, because you're controlling the amount of substrates that enter into the cycle. And then for regulation, just remember substrates promotes, products inhibit. So I believe, yeah, so ADP promotes. Wait. Is that right? Yeah, I guess so. That's right, because just know ADP promotes it, and then NADH inhibits it, because the product is NADH. So yeah, these are the products of TCA cycle, and these are the substrates. Any questions about that? Oh, and just know that calcium is an activator. That's really it for this lecture. Any questions? Okay, ETC. Oh yeah, I just go through them. Because it's going to be about regulation or the poisons. So guaranteed questions on the poisons, and then guaranteed questions on the regulators. And again, it's by feed forward again. So the products of products of this cycle are ATP and NAD plus, and then the substrates are ADP and NADH or FADH2. So anything, those three will promote this. And then just know your poisons, just know what inhibits what. Yeah. So 1 and 2 are separate. So what was it? let's say if you inhibit complex 1, you still have complex 2. If you inhibit complex 2, you still have complex 1. But if you inhibit complex anything down the line from complex 3 forward, you inhibit the entire cycle. And just know about the what was it? The uncoupling transporter. What was it? It was this one. The ATP, this one. No, that's not it. What is it? Oh, this one here. An uncoupling protein. Okay, this is what your brown fat does to make you warm. Any questions? Okay, so reactive oxygen species. Okay, so ischemic cell injury, low oxygen equals low ATP. Low ATP leads to you can't power your sodium potassium pump. If you can't power that, you're not able to excrete sodium from the cell. So there's a high sodium in the cell. Water follows salt, so there's a bunch of water flowing into the cell, and then the cell lice. Everyone got that? Okay. So in low oxygen conditions, HIF produces EPO, so erythropoietin. Erythropoietin makes more red blood cells, and more red blood cells allow more oxygen carrying capacity. Okay, and then when there's high oxygen, you have HIF prolyl hydroxylase, and that breaks down HIF. Okay, so how free radicals damage these things? So in proteins, it denatures them and breaks their bond. No, it just breaks their bonds. For DNA, it causes double-stranded DNA breaks, and then that causes mutations. And for lipids, it's lipid peroxidation, so you're breaking apart the entire lipid membrane. Okay, and then this is the slide that you have to know. So we have a question on the slide. So just know what the enzyme neutralizes is. So superoxide dismutase, catalase, and glutathione peroxidase. So just know for catalase, the two enzymes, for hydrogen peroxide, the two enzymes that neutralize it are catalase and glutathione peroxidase. Okay. So for glutathione, sure. Okay, so for glutathione peroxidase, every time you use a glutathione peroxidase, you have to remake it, and the enzyme that remakes it is glutathione reductase. And to power glutathione reductase, you need high amounts of NADPH, and to make NADPH, the enzyme is glucose 6-phosphate dehydrogenase. And this is your HMP pathway. Any questions about this? And if you ever see fava beans, the answer is going to be glucose 6-phosphate dehydrogenase deficiency. Not for this meeting, but if you ever see fava beans. And then NADPH oxidase. This is what your macrophages use to kill bacteria that it eats. So it makes a superoxide radical. Okay, any questions? Let's see. I'll just do this one. Membrane potential. Okay, so just make sure you know how to do the math equation you did in class. Okay. So make sure, so make sure to know how to do that equation. And then like he could ask you that in reverse. So instead of finding out the charge, you might have to calculate the amount of ions inside or outside the cell. So make sure you know how to do it both ways. Okay, and then for the action potential, so know the stages, know the ion channels that are involved. Did you learn about the ion channels for each one? Yeah, so we'll know the ion channels, and then the directionality of each ion. And then for the states of the sodium, just know which one is in the absolute refractory period, and it's caused by the closure of the activation gates. Again, just know the difference between absolute and relative. Yeah, absolute and relative. So absolute, you can make an action potential. Relative, you can make an action potential, but you need a higher stimulus. And then just know what makes. So just know what makes a fast membrane. So high membrane resistance, low axon resistance. Okay, EPSPs excites, IPSPs inhibits, and these are, we forgot, it's not an action potential, so what's the opposite of action potential? Graded potential. So these are graded potentials. And just know the difference between graded and action potentials. You have to question on that too. That's also good. Okay. So signals that are hydrophilic, they require a membrane. So the receptors are on the extracellular membrane, but they do not require transport proteins that are being carried in the blood. For hydrophobic signaling, because it's lipophilic, its receptor is inside the cell, but it needs carrier proteins when it's in the blood. And then one more thing you need to know is that anything that has a membrane receptor requires secondary messengers. Hydrophobic signaling is usually activating the transcription factor directly, so it doesn't need secondary messengers. And the main secondary messengers are calcium and cyclic AMP and cyclic GMP. Just know that the G protein coupled receptor crosses the membrane 7 times. And just know, again, just know that anything lipid soluble, their receptor is inside the cell, and it'll directly activate a transcription factor. Don't worry about this. If you don't already know the pathways for the G proteins, so GS, yeah, just take a picture. Anyone need more time? Okay, what is this? I guess just know that GTP activates it, GDP inhibits it. Okay, so the ways to inactivate G protein coupled receptor signaling. So this is one way. It's by inactivating the receptor itself. And to do that, you first phosphorylate it by GRK. Then once it's phosphorylated, it allows for arrestin to bind to it, and then it inactivates it. But just remember that the trimeric G proteins itself are able to inactivate itself. Then the other way is to either take in the receptor or break down the receptor. Okay. Cool. Okay, I'll do Na's lectures because she's like a lot of her questions are word of mouth from the tutors, so okay. So make sure you know the weeks for the MBI periods. So all 3 weeks. And then what is it? If she asks you to date a fetus, and then she gives you the date of the woman's last menstrual cycle, you just add 2 weeks. Everyone got that? So if she asks you to identify the age of the embryo, and then she gives you the date of the last menstrual period, you just add 2 weeks. So if it's like her last menstrual period was like 38 weeks ago, wait, no, it was, no, better. If it was 18 weeks ago, the fetus is 20 weeks old, yeah. So when a female is born, her eggs are at prophase 1. When she is beginning to ovulate, she goes to metaphase 2. And then once she decides to have a baby, that's when metaphase 2 completes. So know that the viability of an egg is 24 hours. For sperm, it's 2 days. And just know that the green one here is the most likely site for fertilization in ectopic pregnancies. So in order for the sperm to penetrate into the egg, it first has to be capacitated. And what capacitation means is that the glycoprotein coat is removed. And if the glycoprotein coat, it's a coat, so once coats the sperm. Any questions? When it binds to the zona pellucida, it causes release of acrosomes. Then those acrosomes break down the membrane of the egg, and then it allows it to penetrate. And then once the sperm DNA penetrates, it triggers the cortical reaction, and then that cortical reaction prevents passage of other sperm. Yeah. Okay, weeks 1 and 2. When the zona pellucida is still there, Yeah, when the zona pellucida is still there, that the embryo is in compaction. Wait, is that right? Yeah, that's right. So 1 to 8, it's called compaction. 16, it's called the marula or mulberry. I think that's all you need to know. But like, what was it? Like the broad umbrella terms, so like day 1 to start of week 2, it's the blastocyst. Week 2, start of week 3, it's the bilaminar germ disc, and then week 3, week 4, trilaminar germ disc.
Can you repeat those?
Yeah, so day 1 to start of week 2, it's a blastocyst. Week 2 to start of week 3, bilaminar germ disc. Start of week 3 to week 4, it's trilaminar germ disc.
Thank you.
Okay, just know that implantation occurs at week 1 at the posterior superior wall of the functional layer of the endometrium. And then for these pictures here, make sure you're able to label them. So at the 9th to 10th day, the fibrin plug forms. And this is when the embryo is fully, when it fully penetrates into the endometrium. And if she asks you the question, what stage is the embryo at the formation of the fibrin plug, it's a blastocyst. Oh, and just know that the syncytial trophoblast are what takes it deeper into the endometrium, and then the cytotrophoblast, they're there. They're able to they're able to make more syncytial trophoblasts from the cytotrophoblast. Whatever. Okay, syncytial trophoblast makes HCG. Okay, weeks 3 and 4. I forgot to mention week 5 is the most sensitive. Okay, so make sure you're able to label this picture. Just whatever I highlighted, just make sure you're able to label it. And then this is just a summary of what you have to know. So just know what I did not cross out. So Cyriomelalia, it's yeah, it's there. Loss of mesoderm, and just know that they have renal agenesis. Remember, when you have renal agenesis, you will not have lungs. Okay. The pyramid streak and disappears at week 4, and if it doesn't disappear, you get the sacrocardial teratoma. And just know that this is not cancer. Just know that the nodal chordal plate induces neurulation. And then the nodal cord turns into the nucleus pulposus. So that big flow chart summarizes all these slides. I'm going to skip it. Yeah. Okay, so just know the closures of the cranials and caudal neural cord So cranial usually poses first, and caudal is slightly related. So cranial 25 to 26, caudal 27 to 28. So make sure you're able to label these two pictures on the bottom. So make sure you're able to label those pictures. And then know what they all turn into. Okay, yeah. Any questions about this? So just be able to label it and then know what they turn into. Okay. And just know BMP4 makes your special keratin, so it makes your skin, hair, and nails. Okay, so dorsal pathway for neural crest migration makes your lymphocytes. Ventral pathway makes your peripheral nervous system. And just know that odontoblasts, so your teeth, are made by neural crest cells. Okay, so anencephaly is a failure to close caudal, and then spinal bifida is failure to close. Wait, that's not right. Okay, then what's cranial? Just know that it's prevented by voids. Okay, so your mouth, also known as your oropharyngeal membrane, opens at week 4, then your anus opens at week 7. So know the fetal and maternal part of the placentas. So fetal chorionic plates, maternal residual basalis. Then be able to label this entire picture. So fetal blood is closer to cytotrophoblasts, maternal blood is closer to syncytiotrophoblasts. And just know that fetal and maternal circulation are, they do not mix, so everything, transport relies on diffusion. And septas are cotyledons. I'm not sure if you have to know this, but you have to check for cotyledons when you give birth to the placenta. So you need to know that oligohydra, whatever this is, just know that it's low amniotic fluid. And because there's basically amniotic fluid is baby pee, so if you're not able to make baby pee, that means you have no kidneys, or something's wrong with your kidneys. And just know that if you don't have amniotic fluid, the baby does not have resistance when they're breathing, because the air will flow in the lungs. Less fluid will flow in the lungs, so that's like less exercise for the baby when they breathe, so they'll develop pulmonary hypoplasia. So make sure you tie this with pulmonary hypoplasia. So no kidneys, so no kidneys, no amniotic fluid, no exercise for the baby's lungs, then pulmonary hypoplasia.
Hypo or hyper?
Huh?
You said hypo or hyper?
Hypo.
Hypo.
So no lungs. And then polyhydramnios is excess amniotic fluid. Just know maternal diabetes for that. And it makes sense because when you're diabetic, you eat a lot, so yeah. Okay, and then just know that the connecting stock is 2 arteries, 1 vein. And the way And just know that IgG is able to cross the placenta. And lipid-soluble hormones. So this is inheritance patterns. So you're going to get 2 questions on the pedigrees. So make sure you're able to know them. So autosomal dominant, autosomal recessive, X-linked dominant, X-linked recessive, mitochondrial. And then the other 2 questions will be definitions. Like identify the definition in the vignette. So phenocopies, pletropy, contiguous, locus heterogeneity, yeah, everything here. Any questions? Okay, so second part of cell signaling. Let's see. So nitric oxide, cyclic GMP activates nitric oxide, insulin and growth factors bind to receptor tyrosine kinase. Okay, that's all I have for that. Let's see. So, cholera toxin and pertussis toxin. So, cholera toxin makes an overactive GS, and pertussis toxin make a GI that's not working. So, for cholera, you're having constant signaling, and that causes diarrhea and stuff. And for pertussis, you're not able to turn on the inhibitor, so you'll have overactive secretions. But you just remember cholera, GS, pertussis, GI. Okay. So GQ causes an increase in calcium. So IF3 binds to the ER, causing calcium release. And then the calcium goes back to the IF3 receptor and causes more calcium to be released. And then for this one, so what was it? So just know nitric oxide causes vasodilation. I'll just say just in case he asks you this. So atrial natriuretic peptide binds to an extracellular guanylyl cyclase. Nitric oxide binds to an intracellular guanylyl cyclase. I'm not sure if he's going to ask you this, but just in case he asks you that. Okay, what is this? I guess just know that just know that, wait, no, that's not right. I guess just know that, I guess, you don't even need to know this. Or I'm not sure if you have to know this. Did you go in depth on this? Don't worry about it. I guess just know that MAP kinase activates phospholipase A2. Phospholipase A2 makes arachidonic acid. Okay, so the two types of tyrosine kinase receptors, your monomers and then your dimers. So dimers, they're made of two, monomers are made of one. So something that monomers have to do is that they have to associate with another monomer. Because you need two receptors to activate it. Am I making sense? That wasn't me. Okay, okay, so this is what I was talking about. So this is a monomer, and then monomers have to dimerize to cause activation of signaling. Is this important? Okay, just know that MAP kinase is activated by mitogens, and then MAP kinase pathway promotes progression from G0 to G1, and then just know the order of it, so RAS, RAF, then ERG. And then just know for this pathway, you have to know that the thing that phosphorylates it is the JAK-STAT. It's not the receptor. So what phosphorylates stuff is the JAK. Not important. Any questions? Okay, cell cycle. Okay, so all cancers promote progression from G0 to G1. So cyclins control the cell cycle. So cyclins rise and fall, whereas CDKs remain constant. And just know that cyclin E is what promotes, or is what's synthesized by mitogens. Yeah, that sounds right. And but what's more important is cyclin B, so the fall, and cyclin B promotes anaphase. Cyclin B.
D for mitogens.
Huh?
D for delta for mitogens.
Yes.
B as in bunny. Cyclin B. Huh?
D is for mitogens, B is
Oh,
for the
that's, yeah, it's D, sorry. Okay, so cyclin D promotes G1. Okay. And then to phosphorylate a CDK, you need to phosphorylate it. Wait. To activate a CDK, you need to phosphorylate it. And what phosphorylates it is CDK activated kinase. Okay, and then this is your G2M checkpoint. So if you phosphorylate it twice, you inactivate it, and that's done by V1 kinase. So think of it, V1, you're peeing on the CDK, so you're inhibiting it. And then what removes that extra phosphate is CDC25, and then that will cause the CDK to go past the cell cycle, or past this checkpoint. Okay, and then the other way to inhibit it is when B27, it just hugs it, and then it hugs it, then it inhibits it. There's a better slide for that. And just know that SCF breaks down the inhibitors. SCF breaks down CDK inhibitors. So just know that P53 is the main checkpoint for G1/S. So when there's like DNA damage or too much mitogens, P53 becomes activated, and then P53 makes P21, and what P21 does is to halt the cell cycle. Okay, but the other thing that P53 can do is, can you talk about P16? Does that sound familiar to anyone? Okay, just know that P53 can also cause apoptosis. We talked about that. So fall in cycle B causes a decrease in securin, and it allows for separates to break apart the chromosomes. So fall in cycle B, decrease in securin, activation of separates to promote interphase. Cool. Yeah. Any questions about the subject? Okay, abnormalities. You just need to know that a bar body is an inactivated X chromosome. So if they give you this phenotype or genotype 47XXY and ask you how many bar bodies, it's 1 or 2. That's 1. You get it? It's an inactivated X, yeah. But if it's 47 triple X, it's 2. Okay, for mosaicism, just know that it's just a variation. So variation of cell types. And there are 2 types of mosaicism. There's germline, so it's in your germ cells. And somatic, so it's what you're expressing. So germline could be passed down, somatic cannot. And then just know that for somatic, the earlier it happens, the more variation you'll have. The later it happens, the less variation you have. So this person right here, her somatic mosaicism occurred later. This person here, it occurred earlier. Any questions about mosaicism? Okay. The P arm is the peak arm. And then just know the process of the karyotype. So know the order and know the function of everything you're adding. So phylohemoglutamine, it induces the cell cycle for your white blood cells. Colchicine causes your white blood cells to halt at metaphase, yeah. And then hypertonic saline causes the cells to swell to make the chromosomes more visible. And then trypsin and YAMSA, they cause the banding, and that's what allows you to see it. So again, just know the functions and the order of what you're at. Okay, acrocentric chromosomes have satellites. And just know that these are involved in Robertsonian translocations. And then for metacentric and submetacentric, it's going to be reciprocal translocation. Okay, and just know that 13 to 15 are your large acrocentrics. Then your small acrocentrics are 21, 22, and Y. just know that these aberrations for trisomy and monosomy, they're caused by non-destruction, and as a female gets older, chances of non-destruction increases. So the viable trisomies are 13, 18, 21, X and Y. Trisomy 16 is the most common non-destruction, but this leads to spontaneous abortion. And then just know the only viable monosomy is X. And 69 chromosomes are polyploidy. Okay, and then for the nomenclature, you're just able to, you just need to be able to read it. And then you need to know like what these abnormalities are, like in vignette. So deletion, you'll say you see a shortened chromosome, but all the other chromosomes are normal. Inversion, you see a chromosome that's flipped. Duplication, you see a chromosome that appears double in size, but the rest of the chromosomes are normal. Insertion, you see a large chromosome, but then you see one chromosome that's shortened. Ring, no telomerase. Translocation, or reciprocal translocation. Like, you know about fish? Yes. Never mind, just don't worry about these then. Then for isochromosome, just know that instead of having like picture chromosome, like here's the P arm, here's the Q arm. For an isochromosome, it's going to be P arm, then P arm, and then the next chromosome will be a Q arm, then Q arm. Let's see. So reciprocal is again anything. It's your submetacentric and metacentric chromosomes. Robertsonian only your apocentric chromosomes, and just know that the satellites will become lost. Okay. So the two ways to get Down syndrome. So the one from reciprocal translocation. So that's your. You'll just see a plus 21. You'll see a 47, then a plus 21. And that will be for your reciprocal translocation for Down syndrome. Or wait, your non-destruction for Down syndrome. And then for the Robertsonian translocation, the other way to get Down syndrome, just remember this stereotype. And just know that there's no 10% for getting Down syndrome when they have a Robertsonian translocation. And if it talks about a mom having 5 children who all have Down syndrome, she has a Robertsonian translocation. Huh? A mom who has 5 children who all have Down syndrome, and she's normal, she has a Robertsonian translocation. Okay, so Klinefelter syndrome, we need to know that the problem is with testosterone. There are 2 presenting types. One will be one will be a learning disability, and the other will be infertility. So those are the 2 presenting problems. And just know that what's common to both of them is gynecomastia. Okay. Cool. And just know the filial chromosome, or CML, is an overactive tyrosine kinase. Any questions? Okay. You want me to go through this lecture or no? That's it. I don't, because I don't want to. I'll go through embryo though, because it's kind of dumb. Okay, so first off, long embryo. Okay. So the respiratory diverticulum is the separation of the esophagus with the trachea. The first structure that comes out of it is the lung bud. I think that's all you have to know. Okay. And just know that for this one, tracheoesophageal fistulas, let's see, if she talks about like after you feed a baby, she starts coughing up and becomes cyanotic. Any questions about that? And just know that for tracheoesophageal fistulas, they have common presentations with bacterial, and just know what bacterial stands for. And then know the weeks for the stages for this. Okay. What's important is that type 2 pneumocytes produce surfactant, and if the baby does not, if the baby either has low surfactant or is born too early, when there is no surfactant, they'll get respiratory distress syndrome. Respiratory distress syndrome, and the way to treat it is with surfactant. So pulmonary agenesis, or whatever, pulmonary hyperplasia. The causes of these are oligoamnionic fluid, and then the congenital diaphragmatic hernia. So the two causes of pulmonary hyperplasia, oligohydramnion, whatever, renal agenesis, and then this one, congenital diaphragmatic hernia. Okay, heart embryo. So just make sure to tie in the adult heart structures with the embryological heart structures. So she'll give you like the adult picture of a heart, she'll point at the structure, and then she'll ask you what is the embryological origin of this structure. Okay, just know that vitelline veins will make your blood supply for your GI tract. So it'll make your celiac artery, SMA, and INA. So anything with vitamin, think vitamins, and with vitamins, just think of GI. Whatever. Okay, so make sure you're able to label this picture, and then know what they form. Okay, so truncus arteriosus will make your pulmonary artery and aortic arch. Your bulbus cordis will make the smooth parts of your left and right ventricles. The primitive ventricle will make the rough portions of your ventricle. The primitive atrium will make the rough portions of your atrium. And the right horn of the sinus venosus will make your SVC. The left horn of the sinus venosus will make your coronary sinus. And just know the special names for the, I think in the adult heart they have special names like trabeculate carne, pectinate muscle, so tie that in as well. Just know situs inversus is when everything is switched. Just know isolated dextrocardia is only when the heart switch. Yeah. Okay. Maybe we can talk about that. Okay, just know that the crista terminalis is what separates the smooth and rough portions of the right atrium. So that's inside the heart, and what's outside the heart, it's called sulcus terminalis. Okay, and then know that there are 3 shunts in your heart. So ductus venosus, this will have the most nutrients and oxygen, and it will bypass the liver and go straight to the IVC. Vein valve, it bypasses your right ventricle and goes to your right atrium. And ductus arteriosus, it bypasses your lung flow, it goes straight, it bypasses the lungs, it goes to the aorta. Did everyone get that? Okay. And just know that the ostium secundum is formed by apoptosis. Okay. So your atrial septal defects. So pro-patent frame novell, it's caused by an abnormal fusion of tissue. And then the rest of these guys, it's caused by missing tissue. And if it shows you this picture and asks, what is the chance of this person developing migraines? It's 25%. Oh no, it's present in 25% of people. So 25% of people have this. Okay. Okay, so the heart conditions that will cause cyanosis, they all start with a T. So terrible T's. So tetralogy-transposition and truncus arteriosus.
Is there anything we should know about the acyanotic? Because I remember her mentioning that she would focus mostly on the acyanotic.
Not really. Just be able to know what they look like.
Okay.
And just, like I'm telling you that this is important because in the vignette they'll say this may be cyanotic. So you can instantly cross out these conditions. And vice versa if it was acyanotic. Okay, so ostium secundum defects, the problem is with the formation of the atrial septum. And for the diseases, for these lectures, just know what caused it. So ostium secundum, problem with the formation of the septum. Endocardial cushion defects, problem with the endocardial cushion. Straightforward. Sinus venosus, problem is with the sinus venosus. And she likes asking, she doesn't really like it, but she's asked this before. So she would give you this and ask you how many atriums and how many ventricles does this person have?
We didn't get that study.
So, and vice versa with the other one for ventricular septal defect. And this is just a slide. Pretty much just on the slide. I already talked about it though. Okay, ventricular septal defects. So we need to know that the endocardial cushion forms the membranous portion, and the muscular portion is made by myocardium. And just know that neural cells induce spiraling of the aorticopulmonary trunk. Membranous, membranous VSDs are common. So transposition of brain vessels, it's caused by a failure to spiral. Transpositioning of brain vessels. Okay, a persistent truncus arteriosus. Just know that there is no aorticopulmonary septum. So you have one common outflow vessel. And just know that they always have a ventricular septal defect. Okay, then transposition. The aorta is on the right side, pulmonary artery is on the left side, and what you need to keep the baby viable is you have to maintain the ductus arteriosus, and to maintain it, you give them prostaglandins. So prostaglandins keep it open. And then, yeah, this is what it looks like. Tetralogy of Fallot. The way to remember what caused it is proof. So P stands for pulmonary stenosis, R stands for right ventricular hypertrophy, O stands for overriding aorta, and V ventricular septal defect. The first aortic arch forms the maxillary arteries. Second aortic arch forms your stapedial arteries. Third aortic arch forms your carotids. Fourth aortic arch forms your arch of the aorta. And the right fourth aortic arch forms your right subclavian artery. And this is where your right recurrent laryngeal nerve loops under. Fifth arch does not make anything. Sixth arch makes your pulmonary artery, and the left sixth arch makes your ductus arteriosus, and this is where your left recurrent laryngeal nerve loops under. So yeah, so make sure you know which arch loops for each laryngeal nerve. Just know for the coarctation, it's the typical ribs that will have rib notching. Typical ribs will have rib notching. And then for double aortic arch, just know that it's caused by the failure of right dorsal aorta to regress cranial to T4. Make sure you know that word for word because you'll say failure of left dorsal aorta, failure of right ventral aorta, failure of right dorsal aorta to regress caudal to T4. So just know this word for word. Okay, so vitamin, every time you see vitamin, it's think of vitamins and vitamins, GI tract. So your vitelline artery forms these structures. Celiac trunk, SMA, IMA. Yeah. Your internal iliac artery is directly, internal iliac artery connects with the placenta. And then just know what structure, just know, actually not this one. just know for the pathological veins, just know what caused it. So this one, when the person has no IVC, their hepatic segment doesn't form. The IVC left supracardial vein cyst. The SVC, whatever this one is, isolated left, yeah. Okay, yeah. Okay, so patent ductus arteriosus. To close it, you give them aspirin. So aspirin closes it, prostaglandins open it. Any questions? Okay. Okay, sign in, please.